Okay, hi folks. This is the video to describe the steps we're going to take in our RT-PCR practical class and to explain what's going on at each stage. Again, I recommend watching the practical class overview video first, and don't forget we'll discuss some of the points and some of the content of this video in greater detail in the class itself. Okay, very briefly, what my team have done before the practical is they transfected cells with the different mini-genes or the control plasmids and then extracted the RNA from the cells 24 hours after transfection. And then in the practical class, you'll be handed these RNAs ready to go, and your job is to do three things. First, you'll perform a reverse transcription reaction to convert the RNA into complementary DNA, known as cDNA. Then secondly, you will use the polymerase chain reaction, PCR, to amplify the specific regions of cDNA in your sample. Then finally, you will run your PCR products on an Agarose gel and visualize the bands. Okay, from doing that, and from the final gel image, we'll be able to determine which products are present in each of our samples, and we'll be able to identify their relative sizes or their absolute sizes. From these data, we'll be able to interpret what alternative splicing has happened in each of our different mini-gene conditions. The rest of this video will break down the different steps for this practical. I've decided to incorporate some more sort of the design considerations into this one video, so it's sort of a more complete record, but it's also a bit longer. And because of that, I've broken the video up into the different parts of the experiment to make it easier for your revision. Okay, RNA isolation. Although we did this part for you, I still want you to know what we did and why. Our starting material for the RT-PCR practical is the RNA that has been isolated from our cells. But how did we get it out? How do we purify it? As with many of the procedures we talk about this week, there are different options for RNA isolation. The two most commonly currently used are probably Trizol and some sort of spin column kit. Trizol or tri-reagent is a pretty harsh chemical combination that basically bursts open cells and solubilizes just about everything in them. The tri in the name refers to the fact that different parts of the cells get separated into different fractions of the solution. The main components of the Trizol reagent are phenol and guanidine isothiocyanate. Trizol reagent allows you to perform sequential precipitation of RNA, DNA and proteins from a single sample. After homogenizing the sample with the reagent, chloroform is then added and the homogenate is allowed to separate into a clear upper aqueous layer which contains the RNA, an interphase and a red lower organic layer, and these contain the DNA and the proteins. You can then pipette off the top layer, the aqueous layer, and precipitate the RNA using isopropanol. And the other two layers can actually be collected too. The DNA can be precipitated from the interphase organic layer with ethanol, and the protein can be precipitated from the phenylethanol supernatant from that using isopropanol precipitation. The precipitated RNA, DNA, and protein are then washed to remove further impurities and then resuspended for use in their downstream applications. Trizol performs well with small or large quantities of tissue from 50 to 100 milligrams and large quantities of cells as well from 5 times 10 to the 6th, and works with basically anything, human, animal, plant, bacterial cells, whatever. Trizol also maintains the integrity of the RNA due to how effectively RNA's activity is inhibited during the disruption phase of cells, and it's actually quite good at dissolving the components during the sample homogenization. The procedure is quick and simple, quite cheap, and if you perform extraction well, the total RNA isolated should be free of protein and DNA contamination. But this is something that you will check, and we'll talk about that shortly. Okay, so the other common approach is to use commercially available kits, usually spin columns. So these usually involve some sort of proprietary sample and lysis buffer, the principal component of which is often the same. It's usually guanidine isothiocyanate. And you then follow the protocol which comes with a kit, which usually involves using a centrifuge or vacuum to draw your sample through a column, and that column has a membrane on the bottom that binds RNA. You then wash through the column with a number of different solutions that clears any protein, DNA, or other contaminants, and then finally you elute the RNA from the column using a buffer that will dissolve RNA. Each kit will be designed or will have protocols for different sizes or types of tissue or numbers of cells. So you always need to check that you are working within the range that's acceptable for the kits. With that caveat, kits work really well. They're really simple to use, but they're generally more expensive than the tried reagent. Cost is relative though. If you get good quality RNA and reduce the variability in your assay, you might actually save money by spending more in the RNA isolation step. The biggest variable in most experiments is the starting material, i.e. what you put into reaction. It's kind of pretty simple really. If you put rubbish in, you'll get rubbish data out. So when you're doing experiments, you need to think about what will influence the results and then decide on a quality threshold for whether you should continue or not. In this experiment, we would have cancelled the whole experiment if our cells looked anything other than perfect. We wouldn't have bothered isolating the RNA. That's both before we transfected them and after we introduced our mini genes. We also need to test that our RNA is good enough. Okay, we've got our RNA. There are three things that we need to measure now in terms of our RNA preparations. That's the concentration, whether it's contaminated with proteins or solvents, and the integrity, the RNA integrity, or whether it's been degraded. So let's start with concentration. As you'll see as you go through the protocols, we need to put in enough RNA into our reaction that we don't dilute the other components of the reaction too far. So before you begin, you need to measure the concentration of your starting material. There are different ways to do this. Probably the most common is some form of spectroscopy. And then the other option would be to use RNA binding dyes and then something kind of like the Bradford reaction. The dyes work really simply. They bind to the RNA. The more RNA there is, the more binding there will be, and the greater the signal you're going to get. They're really sensitive, and they're actually pretty accurate. But what they don't do is they don't tell you anything about whether there's any contamination, and they don't tell you anything about the integrity of your RNA. Spectroscopy is probably more common because it's really quick and involves no real cost. It basically involves quite simply measuring the absorbance of your solution at 260 nanometers. No dyes or prep is required, and there are actually quite a few machines that are designed to work with very small volumes in sort of 1 microliter kind of range, so nanodrops, but there's other companies available, which basically makes it really quick. Okay, the same approach, measuring the absorbance at 260, can be used to quantify DNA as well. The calculation of concentration comes from what's known as the Beer-Lambert law, where the absorbance, A, is equal to the extinction coefficient of the type of nucleic acid you're measuring, multiplied by the path length you measured, multiplied by the concentration of the solution in the same units as the extinction coefficient. So basically all you need to do is look up the extinction coefficient of your particular nucleotide, whether it's RNA or DNA, and then rearrange the equation. The accuracy of your absorbance measurement obviously makes a big difference here. For most spec machines, there'll be a range over which the measurements are reliable, so the linear range of the assay. A problem you will run into is that if your RNA is too low concentration, then the measurement won't be accurate, in which case you have effectively just generated a random number, not really very useful. At that point, you probably should consider using a different measurement technique, though possibly if your concentration is that low, it may not be useful anyway. Higher concentrations, too high concentrations, are less likely to be a problem. All you need to do is you just need to dilute your sample down to bring it back within range. Now, your local technical staff should be able to tell you what the dynamic range of your machine is. Okay, apart from the speed and low volume measurements, the advantages, the other advantage of the spectroscopy derived concentration measurements is you also get an indication of whether your solution is contaminated with protein or phenols or not. So let's move on to that, contamination. Okay, as we talked about in the previous video, your RNA isolation procedure involves separating your RNA from the proteins in the cells and involves using solvents to actually do that separation. It uses phenol, uses isopropanol. It also involves cleaning the RNA with alcohols toward the end. Therefore, the final RNA that you get could be contaminated with any of those things, and we want to check if those other components have been carried through to our final solution. Any contaminants could have a negative influence on all the further reactions you do, the RT-PCR steps. Okay, so if you're determining the concentration of your solution using spectroscopy, then there are two easy to do measurements that will give you sort of an early indication of how much contamination you have. And those are the ratio of the absorbance at 260 nanometers relative to the absorbance at 280 nanometers, which will indicate how much protein contaminations you have. And then secondly, the ratio of absorbance at 260 nanometers relative to that at 230 nanometers, which will tell you how much organic solvents you have in your solution. Let's look at the first one, the A260-280 ratio. So let me show you two graphs. The left is pure RNA measuring the absorbance of the same sample at different wavelengths. On the right hand side is a pure protein sample measured over the same wavelengths. You'll see that both solutions absorb light at every wavelength, but they differ in where their respective peaks are. RNA clearly peaks at 260 nanometers, whereas protein has a peak around about 280 nanometers. So what that means is for pure RNA, if you measure the absorbance at 260 and divide it by the absorbance at 280, you get a value of around 2. You can do the same for DNA, and the value actually is around 1.8. However, if you have any protein in your RNA sample, then the absorbance at 280 nanometers will be proportionately higher, and the 260 divided by 280 nanometer ratio will go down, will be lower than 2. Okay, what you have to do as the person who's designing your experiment is you have to decide what is a good enough 260 divided by 280 ratio that means that your sample is appropriate for use in your experiment. So common cutoffs are maybe 1.95 for RNA, 1.75 for DNA, but you might decide that you have to accept less. For example, if you have very old or rare samples where your RNA quality is necessarily going to be worse. The important point is that this is a decision, and that means it has to be written into your methods as it's relevant to the interpretation of your data. Okay, the A230-260 ratio works along the same lines, just this time you know that it's organic solvents such as your ethanol, isopropanol or phenols that are the likely cause of the contamination, and they've come usually from the purification steps. Okay, luckily, conveniently, the numbers are similar. Pure RNA gives you a 260-230 of around 2, and DNA around 1.8. And again, you need to define the threshold that you decided was appropriate for your experiment. Note that an impure sample doesn't necessarily mean that you should be throwing it out. If your concentration is high enough, you might have the option of doing another round of purification to try and remove those impurities. What you should remember is every time you go through the process, the purification and precipitation process, you probably will end up losing some of your sample. So your concentration is going to go down. So the best thing to do is actually be very careful the first time you do it and try and get a pure sample, a good quality sample the first time you do it by following the protocol precisely. So the one thing that measuring the absorbance doesn't tell you is the final kind of important feature of your RNA, and that's whether it's intact or not, whether it's been degraded. To determine that, you need to do something else. The easiest way is to run your sample on an RNA gel. So as far as RNA goes, if you're extracting it from a mammalian cells, you normally will have extracted total RNA, and that means you'll have all the mitochondrial RNA as well as the messenger RNAs in your sample. The mitochondrial RNAs are pretty abundant, and there are two main species that you'll see in every sample. These are the 18S and the 16S RNAs. So when you run a gel, you'll see those two bands as being prominent and a lighter smear of the other material, because none of that is all that is at different sizes, so it's going to spread out more. The mitochondrial RNA bands won't be clear if your RNA is degraded, and basically that's it. RNA degradation is much more likely to be an issue if you're working with any tissue or sample where there was a large delay between the RNA isolation and the purification step. If your sample came, for example, from a laser capture microdissection of archived tissue, or there was a long time post-mortem, or even where you have decided that you wanted to store your plates of cells prior to the extraction, then RNA degradation becomes much more likely and something you always should be checking. And again, in your method section, you need to report how you checked and where your quality threshold was, where you decided it was okay to continue. Note also that freeze-thaw cycles of the RNA you've isolated are also potentially problems. In general, you're much less likely to experience any issues if you extract your RNA at the time of the assay. For cells in a dish, that usually means straight away. So planning your experiments well so that you can remove the gaps between the time that the RNA is ready to be isolated and the time that you purify it will actually reduce the variable in your experiment and reduce that from your experiment. Okay, at this point you've now got RNA, you know how good it is, you know you're ready to continue. So the next little video will talk about the reverse transcription reaction. At this point, in terms of the RT-PCR steps, there are two real choices. You can do a one-step reverse transcription polymerase chain reaction and a two-step RT-PCR. These are pretty much exactly as they sound. In one-step RT-PCR, you use the RNA as the template and perform the reverse transcription and the polymerase chain reaction steps together in the same tube at the same time. Whereas in the two-step reaction, you first perform the reverse transcription, which converts your RNA into cDNA, complementary DNA, and then you use the cDNA as the template in subsequent PCR experiments to amplify your target transcript using the cDNA as the template. In our practical class, we're using the two-step approach, which is probably the most common approach used in discovery science type cases. Okay, in both the one-step and two-step PCR, you have essentially the same things in your tube. You have your RNA as the template, your starting material, and that's come from your reaction, your cells, whatever you're studying. You have a buffer that contains the salts and controls the pH, so that is appropriate for your enzyme. It contains a source of ATP, GTP, CTP, and TTP, the building blocks you need to make your DNA. And this solution is usually called and labeled dNTPs. And you have primers, and finally you have the enzyme. The primer's job is to bind to the RNA and provide the starting point for the reverse transcription reaction. And it's the primers where you, as the experimenter, have to make a decision. Which primers do you need to use? And you've really got four options here. Option one is to use random hexamers. These are exactly like they sound, a mixture of lots of different 6 nucleotide long sequences. These should bind to lots of places in the RNA, and the reaction should therefore start basically everywhere, amplifying every different species of RNA within your tube. Sounds good. The only problem is that all the reactions run from 3' toward 5' end of the RNA. And what that means is you might actually underrepresent the things toward the 3' end, and you might overrepresent things toward the 5' end, or relatively overrepresent. Depends how your question is phrased, but clearly if you have more of the 5' end than the 3' end, this could affect the interpretation of your data. The other thing that would happen with random hexamers is you won't get full-length cDNAs. And this could be relevant if you're intending, for example, to clone the gene, or if you're looking to measure the length of the 3' UTR. So that's where option 2 comes in. And that's to use oligo-DT. So this is, the primer is just a stretch of deoxythiamines, TTTTTTT, usually about 18. And this means that the primer will be complementary to anywhere where you have a long stretch of A's. So why is that useful? Well, at the end of mRNAs, a tail of adenines is added. So mRNAs are described as being polyadenylated. Therefore, an oligo-DT primer will bind to all polyadenylated mRNAs, and then it will work from the 3' end toward the 5' end, giving you the chance to get the whole transcript. However, there are limitations. Firstly, and most importantly, obviously, you will amplify only polyadenylated species, which means that you won't be amplifying ribosomal RNAs, you won't amplify mitochondrial RNAs, and you won't amplify microRNAs. This could be a big deal. Secondly, for long transcripts with complex secondary structure, your reverse transcription reaction might not efficiently convert the 5' end of your transcripts. So the 3' end should be good, but you might not get all the way through to the 5' end. And again, that means that you might artificially change the ratio of 3' end over the 5' end, which would again affect your interpretation. And so that's where option 3 comes in. And perhaps not surprisingly, this is to use a mixture. You get the benefits of both. You use random hexamers to make sure you get the 5' end, and oligo-DT to get the 3' end. Getting good coverage at both ends of the transcript means it's often a good option, but remember that if this isn't what you need, then you might be compromising for no reason. If you definitely want to clone it, you're going to want oligo-DT. Option 4 is to use gene-specific primers, and this is what you're going to use if you're doing a one-step RT-PCR reaction. So basically, you put your RNA in with a primer that for your specific target, the one thing that you want to amplify, the thing that you're interested in, and that's it, nothing else. So in this case, you only amplify what you want. So whereas options 1, 2, and 3 give you greater flexibility, you can assess any transcript once you've made your cDNA. In one-step reaction, you only get the single transcript back. However, you can get more accurate representation of the target copy number. There are less steps along the way. There are fewer pipetting errors and basically less chance of contamination or other sort of mess up along the way. You're doing fewer interventions. So one-step RT-PCR is definitely the way to go if you are screening many samples but only for a small number of genes, perhaps under 4. And for that reason, you find one-step PCR used very widely in diagnostic situations. In contrast, two-step PCR is more common in discovery science, where you have many different transcripts to look at with fewer starting material. So you made a manipulation and then you're looking to see 5, 6, 10 different genes. As with any other point where you are making a decision, the choice of which primer you've used or which primer being used in your RT-PCR step is something that must be included in your methods section because it influences the interpretation of the data. Okay, so now you put all those things into the PCR machine and it will run a cycle at 42 degrees, which will be optimal for the reverse transcriptase to work, and ultimately you'll end up with your cDNA ready for action. In our next video, we'll talk about what you do with that, which is essentially set up a PCR. With your cDNA prepared, or with DNA that you've isolated from cells or tissue, your next stage is to amplify specific regions of that DNA using what's called the polymerase chain reaction. You can classify PCR in two different types. Endpoint PCR, where you're looking to clone or sequence or determine the different splice isoforms produced, or quantitative PCR, where you're determining how much of a specific stretch of DNA is present in your sample. The central principle of both types are the same. You just have to think about a few extra things depending on what your needs are, what your application is. PCRs are relatively simple set up, but there are a few areas where you need to think about things in terms of the design. Each PCR tube will have in it the following things: the DNA template, either DNA, cDNA, or genomic DNA. You'll have a Taq polymerase, which is the enzyme which will be doing the amplifying. You'll have a buffer, which is designed to maximize the enzyme activity. This buffer will often contain magnesium chloride, but if it doesn't, you might have to add it separately, and this is a cofactor for the enzyme. In addition to the enzyme and the buffer, you're going to add dNTPs, which again are the building blocks of the DNA that your enzyme will use as a substrate for its DNA production. And then you'll have primers, and we're going to talk about them in a bit more detail shortly. And then you'll have water to dilute everything to the right concentration. So in terms of design, most of these things stay relatively standard, though there are lots of different Taq and buffer combinations that are designed for specific applications. For example, you might use a different Taq for long or hard to amplify regions than more sort of generally. The area where you'll do most of your thinking is in the design and the optimization of the primer sequences. So just like in the reverse transcription reaction, primers are short stretches, this time of DNA, that bind to specific regions of your target. In this case, you need two. You have a forward primer and a reverse primer. And ultimately, the PCR amplifies the region in between these two primers. So you just need to decide where the primers are going to be placed. And this is where your understanding of your experimental question is critical, as that question will basically tell you how much flexibility you have. The things you need to think about are the location. And for DNA, you can put the primers anywhere. For RT-PCR reactions, you need to design the primers so that they amplify the cDNA and not any DNA that's contaminated your sample. So as you know, genes are made up of exons and introns, and the introns get removed by splicing to make the mature messenger RNA, which leaves behind just the exons. Therefore, if you select a primer that spans the boundary between two different exons, that specific sequence will exist in the mRNA, and therefore in the cDNA, but will not be present in genomic DNA that's contaminated. That's great, obviously, but it also leads to a potential problem, and that's alternative splicing. If an exon is alternatively spliced, i.e. some transcripts have it, some transcripts don't, then your primers won't be useful for generating the absolute quantification of an mRNA if that exon is only sometimes included. Now, obviously, the flip side's true. It could be useful for generating the abundance of the specific splice isoforms. Okay, location aside, next you decide on the length. For quantitative PCR, it needs to be quite short, under 200 base pairs. But for endpoint PCR, it can be as long as you need it to be. But remember, as the length increases, the reaction becomes more difficult, and you'll need to adjust your conditions to consider it. Amplicon length will also influence your cycle conditions. So once we talk about what goes on in the machine, you need to think about that. Next, you have to think about primer length, GC content, and annealing temperature. So what's happening in your reaction is we denature the DNA from double-stranded to single-stranded. Then we reduce the temperature to a temperature that will allow our primers to bind to their target. That binding is down to the specific complementarity of the sequence we've designed within the primer to its target. So when you first learn about DNA, you likely learned that A's and T's bind to each other with 2 hydrogen bonds, whereas G's and C's bind with 3. For your primers, the shorter the stretch of the sequence and the lower the proportion of the G's and C's will translate to a lower temperature being required to allow the temperatures to bind to their target. But here's the thing. Primers will have affinity for other similar but not identical sequences. So what you are looking for is the primer to bind strongly to its target, bind efficiently to its target, and not bind to any of its off-target regions at the temperature you're going to use it at, which we call the annealing temperature. This means that you're looking for some sort of balance. The pair of primers you use should have similar predicted annealing temperatures, and their GC content should be about 40 to 60%, and usually around 18 to 25 nucleotides in length. At the 3' end of each primer, in the direction that your PCR is going to go, it's good to have a little stretch of G's or C's so that the binding is tightest in that region, because you'll get those 3 bonds instead of the 2. So how do you work out the annealing temperature? Well, basically, nowadays there's just lots of software or online tools that you can plug in your sequence into and it'll tell you. But you can get a quick estimate using what's known as the Wallace rule. And that's to count up the number of A's and T's, add them together and multiply them by 2. And then count out the numbers of G's and C's, add them together and multiply that number by 4, then add those 2 numbers together. This rule comes from calculating probes in very specific buffer situations, so it's not really accurate all the time. It can give you a rough approximation, but it's a good place to start. So the question then is, what numbers are you looking for? Well, primers usually work best between 52 and 58 degrees, and you need your pair to be within about 5 degrees of each other. Otherwise, it'd be quite tricky to get optimal binding for both primers at once. If you're planning a whole panel of different primers, I suggest aiming for them all to be close together, as it might make other aspects of your setup easier. For example, you could run all the reactions at the same time in the same machine. Okay, next we talk about self-complementarity and secondary structure. So your primer design doesn't end just by having the right GC content and length. You need to also consider how it folds and also how it interacts with itself and with the other primer. Most online tools will predict whether your primer will fold on itself, i.e. will tell you about secondary structure, or whether it's stick to its pair. It's always best to check this though. In terms of mispriming, you should deliberately avoid stretches of the same nucleotide being repeated, maybe 4 or 5 times, and avoid any repeated sequences like 80, 80, 80, 80. And finally, we need to consider off-target binding. So lots of regions of the genome are conserved, so we need to check that our primer sequence won't be binding to something else in addition to its target. And you do this by performing a BLAST test. Often it'll be integrated into your online tool, but if not, you go to the NCBI website, search for BLAST, and then type your sequence in, and it'll come up a list of complementarity with different DNAs. And what you're looking for is your target to be number 1, obviously it should be, but nothing else to be very good, particularly at the 3' end of your primer. Ultimately, once you've done all this, you'll still need to check your primers. So the first thing you do is you'll do a number of different pilot experiments to work out the optimal annealing temperature, where you get only the right band, the right size, and nothing else. You'll also, if you're doing qPCR, need to check that their efficiency. So at this point, I should say, you may not have to design your primers yourself. You may find them in published work. But even if you do, the next step is to check the primers. With your primers designed and all your reagents mixed at the appropriate ratios, you're now ready to run the reaction. Polymerase chain reactions are done in cycles. Each cycle should double the amount of your target, assuming that your primers are good, your conditions are right, and the primers are efficient. Also that your cDNA is good quality. Okay, before the first cycle, you heat your sample up to 95 degrees for 5 to 10 minutes, and this is to fully denature everything within your sample. This breaks secondary structure and basically linearizes the whole template ready for your primers to bind. After that, each cycle consists of a short step, 95 degrees for 30 minutes, 30 seconds to 1 minute to do the same thing, to break it open, get it ready for binding. You might do this for longer if you're using a long target or have high GC content. Then you drop the temperature to the annealing temperature and you keep it there for about 30 seconds. This allows your primers to bind to their target. And then you raise it to the working temperature of your enzyme, the extension temperature, usually 68 or 72 degrees. And you hold it there for a length of time that depends on the length of your transcript. So it's approximately 1 minute per kilobase of the target. At this point, the TAC does its job, it extends from the primers. And then you repeat. The number of cycles depends on what you want to do and your target abundance. For endpoint PCR, 30 to 35 cycles is probably about right. You should be aware that the enzyme isn't perfect, so the more cycles that you run, the more mutations you'll introduce into your target DNA. And on top of that, at some point you run out of dNTPs. At the end of all the cycles, you usually run a 72-degree step for 5 to 10 minutes to make sure the reaction is fully complete, everything's completely finished. And then finally you drop the temperature to 4 degrees and hold it there until you're ready for action. At this point, you could also take your PCR products out of the machine and put them in the fridge for storage. Okay, so that's it. PCR done. Now you're ready to run the gel. I'm going to do that in the next video. So once your PCR is finished, you're ready to use the PCR products for whatever you need them for, whether it's cloning or sequencing, or in our case, in our practical, we want to check the size of the different products produced from our different mini-genes. And to do that, we've used different primer pairs in each of our samples. Okay, to check the size, we need to separate the products based on their size, and then we need to image them. And to do that, we're going to use Agarose gel electrophoresis. In essence, this is quite similar to SDS page gels we did for the proteins. DNA is negatively charged, so we can run a current through a gel, and we're going to drive the PCR fragments through the gel, and they'll separate based on their size. In this case, the gel is horizontal rather than vertical. It's made of agarose rather than polyacrylamide, and we don't need to use antibodies to visualize our products. We just use a generic DNA binding dye that will bind to everything and then fluoresce. Quite simply, we're going to make up a solution of our agarose at a defined percentage in a buffer. We'll then melt the agarose in a microwave, and as it cools, we'll add the DNA binding dye, and then we'll pour the gel in a casting apparatus, leave it to sit with a comb in place to generate the wells. Then we'll remove the comb, add our samples, add a coloured loading dye that contains glycerol to help our weigh down our samples, and then load the samples into each well of the gel, and we'll add a marker on one end so that we can determine the sizes. And then we'll run the current through the gel. After about 30 minutes or so, our products will separate. We'll take the gel out and we'll image it using UV light. And the image you generate will have one or more bands visible in each lane. And by matching up those bands to the different size and thinking about where our primers are designed, we'll know how big our products are and also basically what they are, which products were amplified in different situations. Basically, that's going to allow you to infer whether full or alternative splicing has happened in each of our different mini-gene cells. So we're going to discuss the interpretation in class, but before I do that, 3 safety points. First, you'll be heating an agarose solution to boiling point, so make sure you're careful when you remove your flask from the microwave that it doesn't bubble over the top and burn you. So make sure you're wearing the heat protective glove. Secondly, the dye that binds the DNA does so by intercalating into the DNA. There is therefore a potential for the dye to have a mutagenic effect. Traditionally, people used ethidium bromide for this, but there was widespread concern about how dangerous, how potentially carcinogenic this chemical was. So it's likely that we'll use one of the modern derivatives that are a little bit safer, but they're still dangerous. So as with all chemicals, treat this with care, especially if you spill anything. And thirdly, we'll be imaging using UV light. UV light causes DNA damage, and your corneas are actually particularly sensitive. So the equipment in the lab will have safety cutouts, so you shouldn't ever be exposed to UV light. But when you're running gels in your own lab, you might be using with an old school transilluminator. So if you're doing that, make sure you're always using face shields, eye protection, and aren't exposing any of your eyes or skin to the light. There are a few things you might modify depending on your experiment. Most obviously is the percentage of the gel. For bigger products, larger products, you'll use lower percentages, and for smaller products, you'll use higher percentages. The ranges used are probably 0.7 to 1% for greater than 2 kilobases, 1 to 1.5 for 0.8 to 2, and 2 to 3% for really small products. But basically you're looking for the appropriate percentage to get the best resolution of your products on your gel. The other choice is the buffer. There are two main options here: TBE, tris borate EDTA, and TAE, tris acetate EDTA. TBE is better conductive medium than TAE, so is less prone to overheating. So you use TBE for long runs. However, the borate is an enzyme inhibitor, so TBE is not a good buffer to use if you'll be isolating the DNA for downstream enzymatic steps, like ligation. So basically use TAE if you plan to use your products for cloning purposes. We're not doing that, this is just for future reference. Acetate actually gives improved separation of large DNA fragments. On the other hand, borate resolves under 2 kb fragments better. So if you're looking for good resolution small fragments, TBE would be better. All this is written down somewhere, so just let you know that you need to Google something. And that's it. PCR, RT-PCR is relatively straightforward. There are many reasons why you might want to do PCR, which includes cloning to express different proteins, sequencing to identify genetic mutations, and quantitative PCR to determine the relative abundance of your target transcript in different samples or conditions. The important point is the basics, the fundamentals of these reactions stays the same. All you need to do is make slight modifications to the protocol to make it better for your application. And so by laying the groundwork in these videos, I hope I've sort of told you what you need to think about so that you can go forth and know what to look up when you're designing your own experiments. High percentage of success in these experiments. Biggest problem is usually contamination along the way. And I never mentioned this as we went through. If you put your own DNA into the sample by having a dust mite or whatever, then you're going to get a product that's not going to be related to our experimental question. So you have to be very careful with all the pipette steps. And that carefulness is really exacerbated by the fact that we're using very small volumes throughout. But that's it. Should be fun. No real problems here. And hopefully we'll get nice clean bands on our gel. Thanks for listening. We're going to go a bit deeper in the lab and discuss things. So if anything has come up that you're not confident with or you have questions, please feel free to ask myself or other people of the demonstrating team. Okay, bye-bye.